gonna ask him if it's
Because I don't know. You have like a strong base.
Wait, which one was that again? Was that before the Civil War? Was that before the Civil War or after? Alright. Thing is, I think John had the text.
Something like that. Just take it right. Just take it and run.
Any questions, anyone? Okay, I understand that Monday's lab had some pretty good success isolating plasmids, so hopefully the rest of the lab sections will have the same success. Yeah.
Those of you who have not yet set up overnights, especially if you have lab tomorrow, you better do it today. If you have lab on Wednesday, you should do it today as well. Or if you have lab on Thursday, you should set up your overnights, because if you don't have overnights, you have nothing to work with in lab.
It just takes a second.
Yes, doesn't take long at all.
Okay, we are in the midst of talking about cancer and talking about the cell cycle, which normally is regulated very precisely within the cell. And if the cell cycle is regulated properly, cell division occurs at the rate it should occur, when it should occur, in tissues in which should occur. Think of the cell cycle in this regard. It's kind of like your car cruising down the interstate. You've got it set on cruise control. You're clipping along. And as long as there are no barriers represented by these checkpoints being kind of raised to stop your progress, you will continue on down the highway, analogous to the cell cycle going through its very phases and then triggering finally nuclear division, cytoplasmic division, the cell divides, and then it's ready to go again. Okay? These checkpoints are very intricately regulated by internal signals that involve the production of various proteins, turning on various genes at various times. Some of the proteins are the protein kinases, molecules called cyclins, and there's this very elaborate orchestration that makes sure that this cell cycle works the way it should work. Okay, if you're driving down the interstate at a set speed, just at the speed limit, right? Got your cruise control set. What can you do to your car to alter your speed when you're on cruise control? You could hit the little button and make it go faster, or could you step on the gas? Okay, and that would increase your speed, so step on the gas. What else can you do to alter your speed besides step on the gas? Hit the brakes, okay? And that's an analogy we're going to come back to in a little bit when we start talking about when this cell cycle regulation mechanism goes wrong. And in some cases, it's analogous to stepping on the gas when you shouldn't be stepping on the gas, or not hitting the brakes when you should be hitting the brakes. And so you continue on through the cell cycle, and the cell keeps dividing when it shouldn't. Okay? In addition to internal signals regulating the cell cycle, there are some external signals that regulate it as well. And one of the things that really does regulate cell division, particularly cells that are growing in tissue culture. Are you all familiar with that idea? We can take human cells out of the body and grow them in kind of complex media that's kind of like sophisticated agar. And under the right conditions, those cells will grow and divide outside of the body in this tissue culture system. But most cells won't grow properly in tissue culture unless you provide some growth factors for them. And one of the growth factors we're going to concentrate on a little bit is this growth factor PDGF. That stands for platelet-derived growth factors. What are platelets? Where do you find platelets? In blood, okay. There are 3 types of major blood cells in your blood. There are the red blood cells, or erythrocytes, which are adapted for carrying oxygen, basically big bags of hemoglobin. There are several types of white blood cells, or leukocytes. And then there are some big interesting cells called megakaryocytes. You don't have to worry about that term. But they fragment into smaller cell fragments called platelets. What role do platelets play in your blood? If you don't have enough platelets, what's the problem? Blood clotting problems, okay? They produce blood clotting proteins. Platelets, as it turns out, also produces other proteins as well. Some of those proteins are involved in a body defense system called inflammation. But platelets also produce a number of growth factors, including the substance platelet-derived growth factor. And this substance is one of the external signals that can cause cells to start dividing again when they're dormant. So what are these growth factors doing? Somehow they are altering these checkpoints, and maybe where there was a checkpoint that was up and there was a blockage in the cell cycle, and under the influence of platelet-derived growth factor, now this checkpoint comes down, and so the cell can move on from the G1 to the S to the G2 and start dividing. So these are really important molecules for us to consider a little bit. Okay? One type of cell that we work with a lot in tissue culture is a human cell type called a fibroblast. Fibroblasts are found in connective tissue in the body. There's a lot of fibroblasts in your body. But again, if you don't provide fibroblasts with the proper growth factors, when you put them in this culture media, they'll just sit there and won't divide. They're not getting the proper signals so that the cell cycle can move on past those checkpoints. Okay? So, here's a little illustration. Here's a petri dish, and somebody's taking some tissue and breaking up with the scalpel and then putting it into a culture media. And then these are cells that the culture media have been supplied with PDGF, and they will divide. But the cells, without PDGF they won't divide. But if they're provided with this growth factor, then they will start to proliferate and form this layer of cells which will cover the whole bottom of that tissue culture flask. They call that a monolayer. And this shows you what these PDGF cells, what these fibroblast cells look like when they're dividing in tissue culture. So the need for growth factors is pretty common. Another thing that cells tend to do is they respond to external signals, which involve cells recognizing one another. Now, way back at the beginning of the semester, we talked about some of the cell surface molecules that allow cell-cell communication to occur. And when cells start growing, if they bump up against another cell, that sends a signal, I'm here, don't crowd me out. Stop growing, okay? There's no need to have more cells. There are plenty of us already. That particular phenomenon is called density-dependent inhibition. And you see that in cells growing in tissue culture. When you first start putting a few of these cells in the media with PDGF, they start dividing and dividing and growing. But eventually, when they start crowding against one another, they start signaling one another, okay, it's time to stop. And they form this monolayer where they form this single layer of cells which kind of covers the bottom of this tissue culture flask. And they don't form these big mounds of cells, for example. They are inhibiting one another from growing. Another external signal that cells typically need when they're growing in culture is they've got to have something to attach to before they'll grow. That is called anchorage dependence. So, here's anchorage dependence. If we have the cells and they are able to attach to the substrate, which in this case would be the walls of the tissue culture flask, they're going to start growing and dividing. But if they can't attach to something, they won't divide. And then they will continue to divide to form this monolayer, but eventually they'll stop growing because of the density-dependent inhibition. You take a few cells out, and these cells are then activated to start growing again. Okay? Removing some of the cells relieves some of that inhibition, and there's also a place down here for new cells to attach. Well, if this is epithelial tissue of your skin, that's a good thing, right? Because if we start getting damage to some areas of your skin, then cells can start dividing again to promote healing. Okay? So cell division is really important in the healing and regeneration process. Now, humans have a fairly limited ability to regenerate tissue. We're pretty good at having cells stimulated to divide and fill in gaps due to damage. But we're not very good at having whole organs, for example, regenerated, or whole body parts. But there are some organisms that do. Okay? If you pull tails off of some salamanders, they will just grow a new tail. And so the regenerative capability of cells and tissues differs from organism to organism. What's interesting about cancer cells is something has happened to them in the transformation process. You remember what transformation means? There are some changes that have occurred within the cell. Their genotype have been altered. And now they behave differently. And one of the ways that they behave differently is they lose this ability to be inhibited by other cells. And they lose the need for this anchorage dependence. So if you grow cancer cells in tissue culture, instead of having this nice flat monolayer, you get all these mounds of cancerous tissue. And you can see here, they're growing on top of one another. Okay? They really don't have a substrate to attach to. They're not inhibiting one another. And I think you can see what this could lead to. This could lead to masses of cells and could become a tumor. So one of the things we want to try to talk a little bit about is what's going on here? Why do those cells lose these kind of critical features? Why do they lose the ability to respond to these external signals, which should regulate their cell division? Cancer cells are sometimes referred to as being immortal. Normal cells have built-in mechanisms, and they involve, including, Jesse asked about telomeres the other day. We didn't talk about telomeres a lot, but they were in the section of your textbook that talked about DNA replication. But there are all kinds of regulatory mechanisms within cells that normally ensure that cells don't live forever. They only divide about 20 to 50 times in culture before they finally stop. Can you see the advantage of cells having a built-in kind of mortality factor? What would be wrong, Jen?
If something goes wrong and it's not harm, they'll die anyways.
Yeah, okay. I mean, if something goes wrong with those cells, then they won't keep perpetuating and passing on that mistake. And what could go wrong with the cell? What could happen to cells as they get older and older?
Your DNA can get damaged.
Yeah, DNA can get damaged. They accumulate more mutations. So at some point in time, a particular cell line has just gotten too old. Okay? Well, there are cancer cell lines, including HeLa cells, that have been growing continuously in culture since the 1950s. Have you ever heard of HeLa cells before? Look, you know what HeLa stands for? It's named for the woman whose tumor they were taken from, Henrietta Lacks. And a lot of research has been done with HeLa cells. And there was a very interesting book written a couple of years ago. The family of Henrietta Lacks was contesting the continued use of her cells because there was a lot of profit being generated in cancer research from the use of these particular cell lines. So that's an interesting question. Who owns those tumor cells? Does a researcher own them because they took them out of a cancer patient? That really raises some interesting bioethical issues. So here's an example, a common example that could change and does happen in your tissues in regard to a change in the cell cycle. So assume that your skin cells are, not dividing. I mean, most skin cells divide, but some will, some won't. You're in kind of a dynamic, steady state. But the ones that aren't dividing because they haven't made it past the G1 checkpoint, typically. So that barrier is up, okay? And that's normal. So the cell's not getting the proper internal/external signals to move that past that particular checkpoint. So it might even slide into that GO phase. So suppose your skin is damaged. Well, one of the things that happens when tissue is damaged is platelets are activated in your blood, and they start producing this growth factor, platelet-derived growth factor. And we'll see how this works a little bit more in the case of cancer. But this particular molecule is a protein that binds to receptors on the surface of cells. And when it binds to receptors, it causes a signaling pathway to be activated. And in that signaling pathway, there is the activation of a G protein, and we'll show you this a little later on in cancer. And that typically involves the activation of protein kinases. These are enzymes that add phosphate groups onto molecules. And that activates other molecules in the sequence. And then typically that activates a transcription factor of some type, which then binds to a regulatory sequence on a gene, and then activates gene expression, and then we start producing some proteins. And those proteins might be the internal signals needed to lower that checkpoint and allow the cell to move through the cell cycle. So cells start dividing. The tissue begins to renew itself and heal. But when healing finishes, the platelets stop producing that growth factor. So now we're not getting the stimulation of the receptors anymore. So this signaling pathway shuts down. We stop producing the transcription factor. And that results in the G1 checkpoint coming back into play again. And cell division stops. So this one crucial regulatory molecule can set off this sequence of events that regulates whether cell division occurs or doesn't occur. The signaling pathways involved with PDGF and other growth regulators are pretty complicated. And this is an example of one normal PDGF signaling pathway that involves the growth regulator binding to a receptor, that activating a signal pathway, and ultimately a transcription factor is produced that causes cell division to kick into place. So these are pretty complicated signaling pathways. Well, what we found out about cancer is that the cells don't respond normally to the body's control mechanisms. One thing that appears to happen with cancer cells is they can grow even in the absence of these growth factors that are normally required to stimulate cell growth. So how can they do that? Well, maybe cancer cells are making their own growth factors. Okay? Instead of having to get them from an external source, maybe the mutation involves them making their own growth factors. Or maybe this receptor can be activated even in the absence of a growth factor. Normally, this has to bind to this for this pathway to be initiated. But in cancer cells, maybe what's happening is this is being activated without any external signal. Or there may be some abnormality in these cells in their cell cycle control system. So maybe they don't need this transcription factor being produced to cause that G1 checkpoint to be lowered so we can go through the cell division cycle. So there's a lot of potential possibilities about what can be going on in cancer cells. Well, one of the early slides that I showed you made this statement, and this is a true statement, that cancer is primarily a genetic disease. So when we talk about cancer genes, there's a couple of major classes of genes that you have to be aware of. And one type of gene is called an oncogene, and a second type of gene is called a tumor suppressor gene. Onc means cancer. So these are cancer-promoting genes. How do you get an oncogene? Well, it's possible you can acquire it from someplace else, like a virus. If you are infected with a virus, some virus will actually insert their genes into your DNA. And there are some viral genes that are actually cancer-promoting genes. But more likely what happens is an oncogene results from a mutation in a normal gene. And those normal genes are called proto-oncogenes. Proto means kind of before, a precursor of. These are kind of the parents of cancer-causing genes, if you like. But a mutation has got to occur to turn a proto-oncogene into an oncogene. Well, isn't it kind of dangerous for us to be carrying around genes within our genome that are proto-oncogenes? That if they're mutated a little bit, all of a sudden become cancer genes? Yeah. So why do we have these? Because many of these proto-oncogenes code for proteins that are involved in regulation of the cell cycle. And if we alter these a little bit, they behave inappropriately. Typically, these oncogenes produce hyperactive proteins that stimulate cell division. These are called gain-of-function genes, and they tend to have a dominant effect. So if you get a mutation in one of your two alleles that turns a proto-oncogene into an oncogene, then you can show cancerous effects in those cells. Okay? Both alleles don't have to be mutated. There is a gene called the RAS gene that we're going to look at in cancer as our example of an oncogene and see what effect it has on stimulating cell division. And it's one of the genes that is involved in this cell signaling pathway that I've got up here. So with our analogy of the car cruising down the interstate on cruise control, converting a proto-oncogene into an oncogene is analogous to stepping on and holding down on the gas. And so cell division is accelerated and continues to go when normal regulatory mechanisms should slow it down and stop it. It doesn't obey the speed limit any longer. Okay? So, here's a receptor in the membrane of a cell. And this receptor binds to particular growth factors from the environment, like PDGF. And when that binds, that can activate a protein, a G protein in this case called RAS. RAS in turn, when it's activated by being phosphorylated by GTP, activates other protein kinases. It results then in the production of a transcription factor. And then this transcription factor interacts with enhancer regions associated with key genes and causes proteins to be expressed that then stimulate the cell cycle. Okay? So basically the mutation results in this hyperactive RAS protein. So RAS was produced normally by a proto-oncogene, but a mutation in the RAS gene produces this hyperactive protein, which maybe doesn't need stimulation by a receptor to then initiate this cascade pathway and cause cell division. Or maybe it does need stimulation, but the response to that stimulation is inappropriate. It overproduces too much signal and causes the cell cycle to start going too fast or not stopping when it should. So it really is like this stuck gas pedal in your car. And cell division keeps going at inappropriate rates when it shouldn't. So, what causes a proto-oncogene to turn into an oncogene? Well, there are several possibilities. One involves the phenomena of translocation or transposition. And this involves a gene actually moving to a new location within the genome. And this is something interesting about genes that we didn't understand until relatively recently. Is that a gene isn't necessarily fixed in one position in the chromosome. It might move to a new location. Or it actually might move to an entirely new chromosome. And when it does that, it might find itself under the influence of a different promoter and a different regulatory sequence than it was before. And so this can alter the rate of gene expression when you move them around. Okay? So here's our proto-oncogene, and it's been moved next to a new promoter, which really actively stimulates the expression of this particular gene. So now this is an oncogene. And it starts producing normal growth-stimulating proteins in excess, like too much RAS protein. Okay? Sometimes genes become duplicated. And so instead of having one copy of the proto-oncogene, we now have multiple copies of that, each producing their protein. And so we produce an excess of protein again right here, more RAS protein than we should. It might even involve a simple point mutation. Maybe that mutation occurs within a control element that regulates expression of the gene. So again, we start producing that growth-stimulating protein in excess. Or maybe it actually occurs within coding regions of that gene. And so in this particular case, maybe the protein that's produced is a little different. And it's hyperactive. It has enhanced activity. Or maybe it's resistant to degradation. Remember, that's one of the mechanisms we can use to regulate gene expression is regulate degradation of the protein that's produced. Now this protein isn't broken down when it should. So there are a lot of genetic changes that can result in the conversion of this proto-oncogene, which codes for normal proteins that regulate the cell cycle, into cancer-promoting genes, which basically, in many cases, overproduce that same protein, which has a disastrous effect on the cell cycle and its regulation. Okay, second important kind of group of genes are tumor suppressor genes. And that name is fairly descriptive. These are genes whose products tend to inhibit tumors from growing and dividing. But actually, these tumor suppressor genes do a lot of things in the cell. There's a particular tumor suppressor gene called P53, which has been studied a whole lot. P53 is sometimes referred to as the guardian angel of the genome. It has a lot of protective functions. If we have mutations in these genes and it decreases the protein products of those genes, that can contribute to cancer as well. So remember our analogy of driving down the highway. A stuck gas pedal is a problem. So are defective brakes. Okay? Tumor suppressor genes put a brake on the cell cycle. They slow it down or stop it. And if your brakes are bad, you're going to continue to barrel down that freeway. Cells are going to continue to divide. One of the things that tends to activate tumor suppressor genes are damage to DNA. So what do these gene products do? Well, some of them help to repair that DNA damage. Okay? What if the damage is pretty extensive? Do you want the cell cycle to all of a sudden start being activated and cells that have partially damaged DNA to divide and pass that on to other cells? No. So sometimes the products of this inhibit the cell cycle so it won't start again until the damage has been repaired. What if the damage is so excessive that the cell really can't repair the DNA? Then some of the products of tumor suppressor genes activate other genes called suicide genes. And those suicide genes cause the cell to self-destruct. And that process is called apoptosis. And that's a really important part of your defense mechanism against excessive mutation is if cells have accumulated way too many mutations, then suicide genes are activated and cells go through this process of apoptosis and they die. So there definitely are times in your cell, in the cells of your body, where it's advantageous for the cells to self-destruct and die. As we've studied tumor suppressor genes more and more, we found out that probably conservatively, about 50% of all cancers involve a mutation in this p53 gene. So this is a very significant contributor to the formation of cancer. So basically what this mutations in this key gene do is they knock out your brakes that normally regulate cell division. So that's serious. So here's what a normal pathway might look like. Here's some environmental insult in the form of ultraviolet radiation, which causes damage to the DNA. That activates genes like p53, which then send a signaling pathway that results in the formation of a p53 protein. This is the active form of the protein. And this is a transcription factor, probably. And so that transcription factor interacts with regulatory sequences associated with genes that produce proteins that arrest the cell cycle. So that checkpoint barrier comes up and cell division stops. But if we've got damage in the DNA and malfunctioning tumor suppressor gene, then we produce a defective or maybe missing p53 protein altogether. Therefore, there's nothing to turn on these genes, and therefore the inhibitory proteins that normally inhibit the cell cycle aren't produced. So again, one small mutation can have a significant effect on whether the cell cycle proceeds or doesn't proceed. So inactivation of tumor suppressor genes like p53 are like having your bad brakes on your freeway. And you continue to barrel down the freeway when you should be slowing down or even stopping. Interference with normal cell signaling pathways like the RAS protein, which has resulted from a proto-oncogene that codes for the normal RAS protein being converted into an oncogene, produces a defective version of this. And we get an overstimulation of the cell cycle. So that's like you're stuck gas pedal and you barrel down that cell cycle when you shouldn't be. Mutations in both of these genes are definitely big players in many types of cancers. So even cancers affect different types of tissue and have different patterns of growth, different treatments. There are some underlying genetic mechanisms that underline all types of cancers. And that hopefully gives us a way to try to attack some of these cancers. So this is a good slide, one you might want to consider for your notes, that shows you kind of the proto-oncogene effect of being converted into oncogenes with the RAS protein, and the effect that defective tumor suppressor genes can have on proper cell cycle regulation as well. In both cases, we get the increased rate of cell division. We either get a protein overexpressed, like in the RAS mutation, or we don't get a proper protein produced, like one that should stop the cell cycle. So we get increased cell division, and we get masses of cells, tumors, forming. Well, in reality, the development of cancer is a pretty complicated process that involves a number of steps and a number of key mutations occurring. So a single mutation typically is not enough to cause a cell to become cancerous. We typically need multiple mutations for cancer to occur. That's one of the reasons why cancer incidence increases with age, as your cells accumulate more mutations the older you get. One of the types of cancer that's been really, really well studied and really supports this kind of multi-step model of cancer development is colorectal cancer. This is a type of cancer that, if it is properly screened, has a very high success for treatment if you catch it early. But if you don't go through the proper screening process, then if this cancer is allowed to progress, it is highly invasive, tends to metastasize, and you have a very poor prognosis. So how do you screen for colorectal cancer? You're too young to have experienced this. Colonoscopies, okay? And colonoscopies are not pleasant. Technologies are happening which are making them less invasive. They're developing little things that are like little pills almost that you can swallow, which are cameras, which will take a picture of your colon as the little pill passes through your small intestine and your colon and is eventually eliminated. They use scopes. But this is something that really needs to be done when you reach appropriate ages and follow guidelines that suggest how often you should have these particular screening processes done, because they're highly effective. The first sign of colorectal cancer is oftentimes a little benign tumor that grows in your intestine called a polyp. And polyp are kind of like precancerous tissues. The cells have been transformed. They've developed some mutations. And they're starting to grow out of control. But if you remove the polyp surgically, then it's not a problem. Now, once you have polyps identified, your tendency to develop more polyps is pretty high. Normal screening suggests that once you reach, about age 50 probably for colonoscopies? Yeah. So, no. About every 10 years for a screening procedure, but if you have a history of polyps, then usually it's 5 years or maybe less than that. You want to keep nipping those polyps in the bud. Fortunately, the scopes that they use for the diagnosis can also be used to surgically remove the polyps. So a physician goes in there and sees some polyps. They can, while they're in there with the scope, actually snip them off, cauterize them, and remove them. Now, in reality, for colon cancer to develop to full-blown metastatic colon cancer, there's probably at least a half dozen changes that have to occur within genes for those cells to become cancerous. And this usually involves at least one active oncogene. So some proto-oncogene has to have been converted into an oncogene. And then usually the mutation or loss of several tumor suppressor genes. So mutations involve both stuck gas pedals and defective brakes. Tumor suppressor alleles tend to be recessive. They tend to be loss of function genes. Remember that I said that oncogenes tend to be gain of function genes and are dominant. So one gene becoming defective here is enough to cause that change. In tumor suppressor alleles, both of your alleles have to become mutated. The one from mom and the one from dad. So, here's our kind of progression of what might happen in the multi-step development of colorectal cancer. So here's our normal colon epithelial cells. They're dividing because these epithelial cells are constantly being shed, and the colon is replacing those with new cells, just like your skin does. But the control is under growth. and they show contact inhibition or density-dependent inhibition. Okay? But if we have the loss of a tumor suppressor gene, this is a pretty common tumor suppressor gene that becomes mutated in colon cancer. It's called the APC gene. Might be other ones though. Then those cells can start growing out of control, and we can form a benign growth, a polyp. Okay? But if in the colonoscopy the surgeon sees that, they snip it out, and that seems to take care of it. But other cells might become sources of polyp growth in the future. If you don't treat this, then if these cells develop additional mutations that results in the activation of that RAS oncogene, now they start producing tumor-stimulating proteins. It's like gas pedal. Another mutation that typically occurs is in a different tumor suppressor gene called DCC. So more loss of function, more bad breaks. So we get a larger benign growth called an adenoma. This can still be surgically removed, but you're one step further on the progression to full-blown cancer. Typically, we need additional mutations as well, and that can involve typically loss of tumor suppressor genes like P53. And now this has developed into a full-blown malignant tumor called a carcinoma. And you can see down here that these cells are invading the healthy tissue. And more likely than not, some of those cells will be released into the circulatory system and they'll be transported to other locations of the body, like the brain or the lungs or the liver, and we'll have a full-blown metastasis. And at that point in time, you've got a real problem now. Now we're talking about chemotherapy. We're past the point where really surgery is a good option because this has invaded all of this other tissue and spread to other parts of the body. So again, the key for colon cancer is really early screening. Well, cancers can run in families. And one of the reasons is that you can inherit predisposition to factors that contribute to cancer, like inheriting some oncogenes from one or both parents, or inheriting mutant alleles of tumor suppressor genes. If you're already born with a couple of mutations, you need to accumulate less of your own mutations in order to have problems. In colorectal cancer, one of the inherited mutations that's fairly common is that APC tumor suppressor gene. So if you get one bad copy and you're born with it, all you need is one more mutation. There are other mutations that have been associated with other forms of cancer besides colorectal cancer. And one that's been in the news a lot are the BRCA1 and 2 genes. They're found in about, mutations in those are found in about half of the inherited breast cancers. And there are blood tests that can be used for detecting mutations in these particular genes. What if your test comes up positive? Well, a woman who inherits one mutant allele, BRCA1 allele, has a 60% chance of breast cancer before the age of 50. If you don't have that, you've got about a 2% chance. That's a significantly higher risk factor associated with one particular gene. So what's happening now? As women are becoming aware of these particular screening mechanisms, and if they know that they have had mothers and sisters and aunts who have died from breast cancer, they oftentimes will say, I want to get that gene test. I want to know if I'm carrying around that mutant allele. And what do some women choose to do if they find they're carrying the allele? Preventive mastectomies, Angelina Jolie. Okay? And that's raised some kind of bioethical considerations as well. Because just because you have a mutation doesn't mean you're guaranteed to have the cancer, but it does increase your risk. Okay? So there are all kinds of bioethical concerns about doctors performing these surgeries on women, even though they have not developed the cancer yet. It gets down to an issue, again, of choice. Other things that can contribute to cancer are DNA breakage. So one of the things that you can do to lower your risk of cancer are to minimize your exposure to agents that damage DNA, like sunlight. Okay? Tanning beds are not good for you. And states are starting to regulate tanning beds, particularly with people under 18 years of old. Chemicals found in cigarette smoke can damage DNA. There are estimates that stopping the use of tobacco could prevent as much as 30% of cancer deaths. So there are some things you can consider doing to try to minimize your exposure. And then there's virus. Okay? We think that virus might play a role in as many as 15% of human cancers. And they do that by donating an oncogene to a cell, maybe disrupting a tumor suppressor gene, or maybe converting a proto-oncogene into an oncogene. This one's harder to control. Okay? But, as I mentioned earlier, some anti-cancer vaccines are being developed for example, the HPV virus, the human papillomavirus. And if you've had the vaccine, then that virus should not be able to replicate. And if it can't replicate, then it can't enter your cells and cause this particular associated damage. Cancer is a really complicated disease, and I think that our goal, which started back in the war on cancer, I think in the 70s, right? To eradicate cancer is probably not going to be a reasonable goal. I think that probably cancer treatment is going to kind of go the way that AIDS treatment is becoming, and try to turn cancer into a manageable chronic disease instead of an acute lethal disease. And there are a lot of treatments that are being developed, and many of these involve sequencing genomes of cancer cells and trying to find out what particular mutation the cancer cells have, and then targeting treatments that seem to be effective on those particular types of tumors. So more personalized type medicine. A lot of research is being done with trying to boost the immune system so that it finds and eliminates cancer cells early on before they really develop into aggressive tumors. We saw in the video about endostatin and angiogenesis that another strategy involves starving tumors by inhibiting blood cell formation, which is needed to nourish those tumors. So there's a lot of really cool things going on right now. We're improving screening mechanisms, but we've still got a long way to go. All right.
I think it should be noted that the vast majority of human tumors are a consequence of our behavior.
Yep.
What you eat, whether or not you expose yourself to things that are known to increase your risk of developing cancer, like sunlight, smoking, food additives, having a diet that's rich or poor in substances that are known to be protective of DNA, vitamin E,
Antioxidants, yeah.
those sorts of things. So don't think of cancer as being inevitable. You can do things to significantly reduce your risk because those things are associated with your behavior.
Okay, so this will represent the kind of required ending point for material for exam number 4. Thursday and Friday, we will talk about virus. And virus, you've got a couple of ways to benefit from studying this material, and that would be to take the additional quiz next Monday, so you can drop 2 quizzes, and then the bonus questions on the exam number 4 as well on the chapter on virus. So that'll finish up Thursday and Friday. Okay?